my research on investigating the mechanisms of neuronal transmission of inflammation to distal neuroanatomical regions, and more specifically, my progress in developing an in vitro platform to study this phenomenon. So, neuroinflammation plays a central role in a number of pathological conditions in the central nervous system. These include things like neurodegenerative diseases, trauma, such as TBI and CTE, neuropathic pain, stroke, and the rejection of therapeutic neuroimplants. And while there has been significant research into managing neuroinflammation, there's still much that we don't know. In particular, there's a very interesting phenomenon in that following a central or a focal lesion in the brain, neuroinflammation appears in regions that are synaptically connected but anatomically remote to that initial injury site. And while there are a number of clinical observations that support this phenomenon, the most obvious examples have been from mouse model studies in which the researchers induced a middle cerebral artery occlusion and follow the histology of the regions of the brain that were synaptically connected. So as you can see in these images, there are a number of excellent examples of significant inflammation appearing in these synaptically connected regions to the initial injury site. And compared to the control regions on the opposite side of the hemisphere of the brain, which show no evidence of neuroinflammation, this supports this phenomenon of synaptically connected neuroinflammation. However, due to the highly complex nature of these in vivo experiments, there have been some confounding results that have limited the extent to which we understand this neuronal transmission of neuroinflammation. Therefore, the goal of my project is to develop an in vitro platform to isolate neuron-mediated transmission of inflammation from other factors found in vivo, and by doing this, hopefully better understand the mechanisms behind it. So on the screen, you can see a schematic of my device. It consists of two cell culture chambers that are connected by a series of microchannels along with an embedded microelectrode array. And the idea behind this is that we can maintain two distinct neural populations in each chamber and then allow them to connect synaptically through axonal projections throughout the microchannels. And we hope by doing this, we can induce a neuroinflammatory response in the source chamber, either through physical or chemical means, and track the progression of neuroinflammation from the source to the target chamber. And by increasing the medium volume in the target chamber, we can create a hydrostatic pressure differential, which will create flow from the target to the source chamber that will prevent the diffusion of any pro-inflammatory cytokines and mediators from confounding our results. So in order to quantify the extent of neuroinflammation in our platform, we're going to look at three major factors. The first being the morphology of the cell cultures themselves. The second being any change in neural activity via electrophysiological recordings from the microelectrode arrays in here. And finally, the change in any pro-inflammatory cytokines or neuropeptides, either through biochemical or electrochemical detection. And we hope by kind of looking at the interplay between these different factors, we can gain a better understanding of the mechanisms behind the neuronal transmission of neuroinflammation. For example, if after we induce a neuroinflammatory response in the source chamber, we see a significant increase in neural activity throughout the platform, this may indicate that excitotoxicity plays a significant role in this phenomenon. So now I'm going to talk about my progress towards the development of this platform itself. I'm going to start on a more biology side in the development of a cell culture model that can better recapitulate neuroinflammation in vitro, and follow it up with an engineering talk about the actual platform development and optimization. So, in vivo, neuroinflammation is a highly complex and interconnected process involving a number of different cell types. However, the three primary ones are shown on the screen, including neurons, microglia, and astrocytes. And while microglia and astrocytes are the main neuroinflammatory cells that respond to extracellular cues and produce neuroinflammation, they respond to different stimuli very differently. In particular, if we see here, microglia respond very strongly to LPS or extracellular glutamate and produce a strong neuroinflammatory response, while astrocytes typically do not. Therefore, in order to produce a robust neuroinflammatory in vitro model, it's important to be able to culture all three of these cells together. However, one of the major obstacles in producing this cell culture is that microglia and neurons tend to have very different cell culture media types, with microglia typically requiring a high amount or a high concentration of serum in the media, which tends to be very neurotoxic. However, due to the nature of our experiments and the design of the platform, it was necessary to produce this robust neuroinflammatory model. So in order to overcome this, we did a deep literature review and found an example of a serum-free media that could support isolated microglia. And this consisted, in addition to the typical media factors, two different cytokines, TGF-beta and IL-34, along with a high concentration of cholesterol. And when we looked at this, none of these seemed to negatively impact the growth of neurons and astrocytes. So what we decided to do was spike our typical neuron and astrocyte co-culture media with these three additional factors. And we found that we were able to successfully culture all three of these different cell types together. So you can see in this top image, we can see the microglia stained with IBA1 in orange, along with the astrocytes stained with GFAP in green, and the neurons stained with beta-tubulin in red. So all three cell types are present in the culture media, versus down here in the co-culture, we don't see any staining of the IBA1. Additionally, we wanted to quantify the number of total microglia within the cultures, and we found that it was approximately 10%, which is a physiologically relevant number of cells. And finally, on this front, we wanted to ensure that not only were the microglia present, but they were physiologically active. So we spiked both our triculture and our co-culture with LPS, which I previously said only microglia respond to. And in this case, we measured the change in caspase-3 activity, which is an indicator of apoptosis and, by extension, neuroinflammation. And as you can see here, the co-culture showed no increase in caspase-3 activity, while our triculture showed a significant increase in caspase-3 activity, almost matching the star scoring positive control. Additionally, when you look at the histology itself, we see a significant increase in astrocyte GFAP expression, which is another hallmark of neuroinflammation. So now I'm going to switch gears slightly and talk about the actual design and fabrication of the platform itself. So the platform was fabricated using pretty traditional technologies used for organ-on-a-chip. Briefly, it just consists of using photolithography to make a master mold, over which we poured and cured a PDMS, which is a biocompatible polymer. And once that was cured, we affixed it to a glass slide to form the final device. And over here you can see an example of the device. It's just loaded with green food coloring in order to be able to see the cell culture chambers a little bit better. And down here we have an example of the master mold highlighting the microchannels themselves. And finally over here, this is a histological image of a day in vitro primary cortical cell culture grown inside the device. And you can see it's very healthy with a large number of neural processes, along with a large number of axons growing into the microchannels themselves. So one of the first things we wanted to optimize was the channel dimensions themselves, because we wanted to limit the number of astrocyte or microglial processes that could enter these channels, as that could potentially confound our results. And as we can see, by decreasing the width of the microchannel, we were able to limit the number of astrocytes that enter the channel. And we believe this is because astrocytes require a neural process to grow over before they can actually extend their own. And we thought by limiting the cross-sectional area, we could perhaps physically prevent the astrocytes from growing within the microchannels. And what we saw here, so now we're trying to actively decrease the size of these microchannels even further, and we're pursuing a couple of designs with even smaller channel widths down to 1 to 2 microns. And finally, we just wanted to validate the two major tenets of the device, the first being that the target chamber can be chemically isolated. So in this case, what we did is we incubated the source chamber with a high concentration of the fluorescent molecule fluorescein, and tracked the fluorescence readout in the target chamber over a period of 7 days. And so due to the hydrostatic pressure differential, we saw no diffusion of any of the fluorescent molecules from the source to the target chamber. However, at day 7, we stopped maintaining this pressure differential and allowed the two volumes of the wells to equilibrate, and then we began to see diffusion of the fluorescent molecules. Additionally, we wanted to look at the synaptic connectivity between the two different neural populations. So in this case, we loaded each side of the chamber with the calcium indicator dye, and then spiked the source chamber with 2 millimolar of KCl in order to depolarize that side, and then looked at the fluorescent readout in the opposing chamber. And once again, we saw an increase of fluorescence, indicating that the cells were depolarizing in response to the addition of KCl in the source chamber. So, in conclusion, we were able to develop a triculture protocol that was able to sustain all three of the major neuroinflammation cell types, neurons, microglia, and astrocytes, to better model neuroinflammation in vitro. Additionally, we designed and optimized a cell culture platform capable of supporting 2 cell cultures that are chemically isolated but synaptically connected. And the next steps for my project itself will be to design and fabricate the microelectrode array platform underneath this, the platform we described, to take electrophysiological recordings. And finally, it'll be to use this new platform to study the actual central objective by inducing a neuroinflammatory response in the source chamber and monitoring the changes in the target chamber to gain a better understanding of the mechanisms underlying neuronal transmission of neuroinflammation. And with that, I'd like to thank my lab group, in particular my PI, Professor Erkin Shaker, along with our collaborators in molecular bioscience, Professor Pamela Line, her recent graduate, Dr. Sanjay Sethi, and her postdoc, Brianna Morgan, and along with our funding sources, in particular the UC Davis DEB program. And with that, I'd like to thank you for your attention and be happy to take any questions.
That was a great talk. Thank you.
Thank you.
How are you designing the microchannels? Are you limiting their sizes?
So that's a great question. I'm using photolithography, so it's like the technology used to make integrated circuits. So essentially we have a polymer that's photoactive, and then when we shine light through kind of like a mask, it cross-links that polymer, and we can get down to 1 to 2 micron dimensions.